Hello, I am Sandy Deans. I'm the Director of the Genomics Quality Assessment Scheme, GenQA, and I'm delighted to welcome you to the GenQA Focus on Molecular Pathology webinar. We're going to present the first pilot EQA for the detection of NTRK gene fusions, which was developed and delivered within a very short timescale, just a few months earlier in 2020. And this was in response to the licensing in England and other countries of the pan-cancer drugs targeted to NTRK gene fusion tumours. It's really important to ensure and evidence high quality testing in the field of oncology when clinical management and treatment options are directly linked to testing results. Next slide, please. So today we're going to cover an introduction to the GenQA Molecular Pathology EQAs. So just a summary of why EQA is important and how we deliver these EQAs for the molecular pathology testing. I'm absolutely delighted to welcome Helen Scheltz from Manchester as a speaker today. Helen is the lead cancer specialist scientist at the Northwest Genomic Laboratory Hub in Manchester and has been instrumental in providing scientific expertise to the GenQA NTRK EQA. We're also going to discuss the results from the NTRK pilot EQA, as I said, that was delivered this year, and then we will have a question and answer session. As we've got so many people on the call, please can I ask you to mute yourself? And if you would like to ask a question, then please enter them into the question box that you can see on your screen, and we shall address them during the question and answer session. I'll now hand you over to Dr. Jenny Fairley, who is the GenQA Deputy Director and manages the molecular pathology EQA schemes. Jenny, over to you now. Thank you.
Thanks, Sandy. As Sandy has mentioned, I'm going to give you an introduction to the GenQA Molecular Pathology EQAs. So, first of all, just by starting who we are as Genomics Quality Assessment. So we're part of an umbrella organisation, UK EQA, which is a consortium of other EQA providers, and UK EQA stands for UK National External Quality Assessment Services. And all of us work together in some respects to help to ensure clinical interpretation is accurate, reliable and comparable wherever they are produced. UK EQA has over 50 years experience providing EQA across biological disciplines, and this includes 25 different specialist centres across the UK. The majority of our EQAs are ISO 17043 accredited, so that is the accreditation, international accreditation for proficiency testing, similar to that for laboratories of ISO 15189. And despite our UK name, we actually have participants all across the world. We're a charity organisation, which means that all our EQAs have to be self-funding, non-profit making schemes. So we can't, anything that excess money we have, we go on to develop new EQAs. So we're always trying to help ensure our participants have their needs met by us. And GenQA has been offering EQA since 1982, and now we offer over 100 EQAs in genomics and pathology testing, and we also have some clinical genetics EQAs. We've got a training and competency tool, GTAT, which we are continuing to develop very actively at the moment. We're based in Oxford and Edinburgh, and now we're the largest genomics EQA provider. And the map behind this slide just shows you all the green shaded countries are where we have laboratories which participate in some of our EQAs. So as you can see, we are very much worldwide. So what is EQA? Well, EQA is external validation of the quality of a service according to international standards. And what that does for the laboratory is provide it with an external measure of the standards of the service. And that that can be used by clinical users, subcontracting laboratories, commissioners, your research and development partners, and your clinical trial partners, to show that your service is of a high standard. EQA helps to improve quality by advising on the analytical and interpretive laboratory performance, and we promote quality across national and international borders, so that results produced in one country should be equivalent to all other countries. There shouldn't be national or international differences. And we try to provide educational and interesting clinical cases and workshops for our participants, such as this, or webinars such as this. So, as Sandy alluded to, I am managing molecular pathology EQAs, and we've been running actually molecular pathology EQAs for 12 years now. We started this, as this timeline demonstrated, we started way back in 2008, offering EQA for gastrointestinal stromal tumours, which we still offer. Then in 2009, that was joined by a KRAS testing EQA for colorectal cancer. In 2010, we also introduced EGFR testing in non-small lung cancer EQA, and at the same time, we offered an MSI, like satellite instability EQA, which was based on testing DNA. This was followed by BRAF and melanoma, so we're looking at obviously biomarkers involved in drug selection with many of these EQAs. And then we also ran our first panel EQA in 2012, which was part of the CRUK Stratified Medicine project, which looked at a number of different biomarkers and different tumour types. In 2013, we made the colorectal cancer, lung cancer, melanoma EQAs, all part panel EQAs, so not just looking at one gene, but multi-genes. We then introduced a sarcoma EQA for RT-PCR, which was followed up in 2015 by the FISH equivalent of this. And we've also run, originally when the NHS England 100,000 Genomes project, we set up EQAs to look at DNA quality and quantity, and they've been continued when we look at various tissue types. So we look at fresh frozen FFPE, blood and saliva DNA extraction, and DNA quality as well. We've set up an EQA to look at molecular tissue identification, so to determine whether two bits of tissue come from the same patient or not. It's from FFPE. And then we started to introduce new biomarkers into the lung EQA. Because these were still quite experimental at that point in time, we set that up as a separate EQA in 2015. And then 2016, we expanded MSI to include tissue, and also set up a BRCA ovarian EQA, and then that was when we introduced an NGS somatic EQA. The DNA extraction from fresh frozen was actually introduced in 2017, and then we also looked at BRCA ovarian interpretation following on from the BRCA ovarian testing EQA. We also introduced a DNA looking at testing and circulating tumour DNA in 2017. 2018, we expanded the MSI EQA to look at mismatch repair, so NLH1 promoter methylation, BRAF, were parts of that. And then in 2020, we've also been involved in setting up some new EQAs, and we revisited our lung and colorectal EQAs to expand the repertoire tests. So we merged our additional lung biomarkers EQA with the lung EQA, and now we test. Now we offer labs with the possibility of EQA for 9 different genes in lung cancer, which includes things like ROS and RET, in addition to ALK, EGFR, and the ones we have been offering for a number of years now. In colorectal cancer, we also included MSI testing as part of the colorectal cancer pathway. We introduced an EQA for BRCA testing in prostate cancer, and are part of a consortium involved in looking at a pilot for human mutation burden. In January, we'll be sending out our cases for our new somatic variant interpretation EQA, and what we're going to focus on today is NTRK, so that was run first this year. And so as you can see, we're continually developing new EQAs depending on the changing clinical picture. It's quite important for us to make sure that we pick up on any new drugs that are coming out or tests that are required, because you need to perform EQA. So the format of our EQAs for tumour testing, solid tumour testing, is we use FFPE material, and we usually use real material, real patient material, and we select a particular mutation for an EQA. We identify a sample where there's enough material available and the consent is appropriate. And for the majority of our EQAs, we require more than one block from the same tumour, or we need to have a tumour big enough that we can make it split it into more blocks as well. We prepare the sections for the EQA, and we have validating laboratories who test the sections independently to ensure that the result is what we would expect it to be. So one laboratory tests the top of the block, the sections that are cut from the top of the block, and the other laboratory tests the sections cut from the bottom of the block. Where possible, we try to use two different methods. And then during the EQA distribution, we actually monitor each section sent to each laboratory to check for any issues that might be with heterogeneity. So we can tell exactly which block which section each lab got. We can look at the labs who receive the sections immediately prior and immediately after them to see if they get the same results. And that way rule out any chance of heterogeneity. So we aim to assess the whole process involved in the clinical service. So we're looking at sending out validated tumour samples where we know what the results should be. And we send them out as rolled sections, slide mounted sections. We give the laboratories the opportunity to do an H&E stain and assess the tumour content, but then test a rolled section. And that should really follow whatever the lab does as their usual process. The labs then obviously also get information on the patient. This is fictitious names and dates of birth, they're not real, and we give them an EQA block number and a little bit of a clinical case scenario. We then ask the labs to test according to their routine protocols. So we're asking you to test it according to how you would test a normal clinical sample, not treat it as special because it's an EQA sample. We want you to report it in the context of the clinical case provided using normal reporting formats. And so I'm now going to pass you on to Helene, who's going to talk to you about testing for NTRK fusions.
Thank you, Jenny, and thank you for asking me here today to talk about NTRK fusions and testing that's performed in the laboratory in Manchester. So first, just a brief background into NTRK fusions and how they form. So NTRK fusions are the fusion of the 5' end of one gene fusing to the 3' end of the NTRK 1, 2 or 3 gene. The specific and important protein domains are the kinase domain and the ligand binding domain in the NTRK genes. And then they usually fuse to a promoter region of another gene, which causes constitutive activation of the NTRK gene. Next slide, please. So there have been numerous fusions described in the 3 NTRK genes, most in NTRK 1, but numerous ones in NTRK 2 and NTRK 3. You will notice that they've been described in all tumour types. They are higher prevalence in certain tumour types. Some paediatric tumours and GISTs are some of the tumour types where you more frequently see NTRK fusions. However, they have been described in many different tumour types. As well as the ones that have already been described, we know that there are frequently novel fusions with NTRK partners, which does create problems for both testing and analysis and interpretation, which I'll come on to. Next slide, please. So there are a number of testing options that can pick up NTRK fusions. Immunohistochemistry, FISH, there's reverse transcriptase PCR, and next generation sequencing technology. So I'll go into these in a little bit more detail. Next slide, please. So immunohistochemistry, it's a routine pathology test which is quick and relatively cheap. It directly assays the protein levels, so you can actually see an increase in protein of the NTRK genes. It's a direct test of the NTRK rearrangement. It's an indirect test of the NTRK fusion rearrangements, so it can result in false positive and negative results. And the analysis can be subjective, and it also doesn't give any information as to the fusion partner involved in the gene rearrangement. Alternatively, you can use FISH. Again, it needs expertise and the analysis is subjective. Again, it leaves the fusion partner and gene unknown. It only picks up the NTRK side of the fusion, and it doesn't actually provide any evidence of pathogenicity for the fusion. There's no assessment of the protein level, and as you can't determine the fusion partner, you don't have any additional information on whether that fusion would be oncogenic and clinically important. Next slide, please. You can use reverse transcriptase PCR. This is a highly sensitive and accurate assay. However, it does rely on you knowing both fusion partners involved. Because it's so sensitive and accurate, it can be used on RNA extracted from FFPE samples, although this does create its own issues in that the RNA extracted can be quite degraded and it's not always of high quality. Again, it's not applicable where there's an unknown partner. It's only relevant if you know both sides of the fusion. And moving on to NGS technology, this again is a highly sensitive and accurate technology. It can detect different variants and also can be used up to secondary mutations, which could be important for drug response resistance, as you can look at single nucleotide changes in addition to the fusions. It's able to detect unknown partners, so can pick up the novel NTRK fusions, which we think is going to be very important with these NTRK fusions because we know there are so many novel rearrangements. It isn't a direct assessment of the protein levels, therefore any fusion detected does need careful interpretation to determine whether it would be oncogenic and clinically important. Again, it relies on RNA, which is extracted from FFPE samples, and the quality of this RNA can create problems within the lab sometimes. This has been the recommended method for the Cancer Drug Fund Managed Access Agreement because of its sensitivity and the ability to pick up all fusions despite the partner. Next slide, please. So how do we assess gene fusions using next generation sequencing technology? We start off with an FFPE sample, either biopsies, resected tumours. We take the material from the block and we extract the RNA from it. We then create a library preparation of RNA. From the RNA, it's reverse transcribed to cDNA and then amplified for a custom target panel of primers, including the NTRK genes, as well as some well-known fusions. This is then sequenced. We then analyse the data and can determine if any fusions are present. Next slide, please. The NTRK analysis of this data, we take the sequencing, we map it back to the reference sequence, the human genome, and then we look for areas where there's a break in the sequence which corresponds to two different genes next to each other. You can see here that you've got the NTRK3 on the right hand side, NTRK3 exon 15, and it's running on into ETV6 exon 5. This is a well-known fusion, and you can see the sequencing reads show that there's a fusion here between these two genes. Next slide, please. We are able to analyse the breakpoints using the next generation sequencing data and assess whether they are likely to be oncogenic, whereabouts the breakpoints are in each gene, whether this corresponds to a potentially oncogenic fusion. Next slide, please. The other main advantage of next generation sequencing is you can analyse, it's high throughput and you can analyse large numbers of samples fairly quickly. This is an example output of a next generation sequencing RNA fusion panel. As you can see, you get a number of calls in an Excel format. You can quickly identify those that are relevant and real fusions, and therefore only need to look in detail of those that pick out the fusions, and therefore you can quickly screen for any fusions that are relevant in a large number of samples. Next slide, please. So just a little slide on sample requirements. It is important to maximise the RNA sample that we get to get the best possible result. So we need to make sure that there is a suitable neoplastic cell content within the block. This is generally regarded and during validation shown to be around 20% neoplastic cell content, gives you good sensitivity for detecting these fusions. The sections ideally need to be mounted, but not, then ideally the sections need to be cut, but they're not exposed to any heat. So preferably curls are required for RNA extractions. These are sent to the laboratory where they undergo next generation sequencing, and then the report is issued. Next slide, please. So as the license for NTRK fusion drugs came into the UK this year, a group was formed with the GLHs around England and NHS England to put together some best practice reporting guidance. NTRK gene fusions are a predictive biomarker for treatment with NTRK inhibitors. We have looked at the guidance as to how to report these and some criteria for reporting. It must be you must be aware that not all fusions are actionable. Not all will create an oncogenic potential and create an increase in protein and therefore be targeted by the drugs. So a number of things have been highlighted as flags for potentially fusions that should not be reported or require additional exploration before reporting. Fusions should be predicted to maintain an open reading frame at the breakpoints at the junction. So any out of frame predictions should be reported with care or not reported before further investigations. The breakpoints need to ensure that the full tyrosine kinase domain of the NTRK gene is included, and need to ensure that they do not remove the ligand binding domain either. So we need both those domains included in the fusion. Often with these predictions, there may be further work required, and it may be that additional evidence is required for these fusions that could be attained by potentially IHC or FISH. Looking at expression ratios, it's sometimes possible to use the NGS RNA analysis to look at the expression of the protein, and also ensuring that there's a forum for further discussion within the community to look at rare fusions and see if they're reported elsewhere. It's interesting to note, and probably quite important to remember, that NTRK fusions are typically mutually exclusive of other common driver mutations, particularly in the MAP kinase driver pathways. And some of the most common NTRK partner genes are listed here: TMP3, LMNA. So there are some common ones, but please be aware there are also many novel gene fusions. Next slide, please. So thank you for listening, and I'll take any questions now or at the end.
Thank you, Helene. I think we'll take them at the end. There's some coming in through the questions box. So again, if we just say, encourage you to submit your questions and we can address them. There's some coming in about EQA as well as the NTRK specific information you've just given. So thank you, Helene. And I'll pass over now to Jenny, who's going to give a summary of the results of the NTRK pilot. Thank you.
Thanks, Sandy. And thanks, Helene, for really comprehensive overview of NGS testing, in particular with NTRK. So we sent out samples for the NTRK EQA in June of this year, and we published the results a few weeks ago to our participants. We had 50 laboratories who registered for EQA from 21 different countries, and that's represented in this graph here. And what you can see is this is very much across the world, labs within Europe, and also outside in Asia and Americas. And of the 50 participants who registered, 41 submitted results. 7 of these laboratories that didn't submit results withdrew from the EQA and provided us with a reason for it. And the main reason why labs withdrew was that the test is still under development, which is particularly surprising, as we know this is very new test in a lot of laboratories. And then 2 laboratories did not submit results and they didn't give us a reason. But as this is a pilot EQA and we don't put any performance on it, then this doesn't have any impact on the laboratory. So the format of the EQA was that we sent out 2 cases, and these were FFPE, and we offered the laboratories a choice of rolled sections, slide-mounted sections, or one slide mounted section plus rolled sections, and that enables the labs to do H&E and tumour assessment if that's part of their normal procedures. We provided a case scenario for each case. We had asked that this EQA would be for NGS or RNA-based testing, so wasn't really designed to look at immunohistochemistry fish. And laboratories were asked to submit the reports in their usual reporting format. We only assessed the genotype and the scores of this, but we did provide some feedback on the interpretation. We didn't provide, there was no per performance monitoring applied, as I mentioned, because it's a pilot EQA, so it's the first time we've run. Very much a chance for us to make sure that EQA is. So the first case we sent out was a pediatric patient. So we gave him some case details. So he was only 2-year-old with a left-sided abdominal mass with extensive peritoneal involvement, and it's a clearly congenital infantile fibromyalgia and NTRK inhibitor therapy was being considered to treat a non-resected tumour. The results of this case were that there was an NTRK fusion present in the samples we sent out. It was an ETV6 NTRK3 fusion, and it was between ETV6 exon 5 and NTRK3 exon 15, which is actually one of the fusions that Helene showed you earlier in the talk. We would expect that participants would be able to detect the fusion in this case, and they'll be able to recognise that this is a known NTRK fusion. This is one that's been identified commonly before. This fusion is actually consistent with the diagnosis of infantile fibrosarcoma, but labs should also be aware that it's also been reported in a wide range of other tumour types, so it doesn't actually prove the diagnosis of the infantile fibrosarcoma, but it is consistent with it, and it's good to include this in the report. And also, laboratories should state that the NTRK fusions are predicted biomarker for treatment with an NTRK inhibitor. So the results for this case 1 were overall very good, in that all the laboratories which participated reported the presence of an NTRK fusion, and most of them did describe this correctly, although one laboratory reported it as being between exon 5 of ETV6 and exon 16 of NTRK3 rather than exon 15. As this was a pilot EQA, we didn't actually deduct any scores, but moving forward, that would be something that would be an error within EQA. Helen mentioned that doing NGS, you should be able to detect where the break point is in a fusion. Although it has to be said that not all laboratories doing this EQA did use NGS, and we'll come to that a little bit later. So the majority of laboratories did report the break point, and that was 34 laboratories, so 83% of them. 4 laboratories used technology that couldn't detect the break point, so they couldn't report it. And then there were actually 3 laboratories, there were 4 laboratories that didn't report the break point, but the technology should have been able to. So they should really be reporting the break point if they can. And then there were 3 that used technology that couldn't detect the break point, so we would expect them to report it. One key thing that we did notice is that nearly a quarter of laboratories didn't include reference sequences on their reports, even then when they report a breakpoint. So we really need the presence of a reference sequence to tell which part and which exon you're actually reporting as being in the breakpoint. Without a reference sequence, it's quite meaningless. So there were 32% of laboratories actually used some kind of nomenclature, either HGVS or ISCN, to describe results. As this is a pilot, we didn't mark this, and we'll come on to the HGVS nomenclature a little bit later because this is quite a new thing. What was quite striking was that I mentioned that the fusion in this case is an ETV6 NTRK fusion, and more than a quarter of the labs didn't mention it was consistent with the diagnosis of congenital infantile fibrosarcoma. So this is that information which could be important for the pathologist or oncologist reading the report. And then there was one laboratory didn't mention about the fact it was a predictive biomarker. This laboratory did mention about congenital infantile fibrosarcoma. Case 2 was an older patient with a GIST. So, and it was wild type GIST, so the child was now looking has recurrent disease, and it's not responding to tyrosine kinase inhibitors, so they're now considering NTRK inhibitor therapy. And there was no NTRK fusion present in this case, so we would expect that the participants would recognise that there was no NTRK fusion present. They would say something about the limitations of the assay they're using, because it's important to know what you can and can't detect. And that as the patient has no NTRK fusion detected, the result provides no indication that NTRK inhibitor therapy would be appropriate in this case. Again, the results of this were good. So all laboratories that reported the correct result when they reported the results of NGS RNA-based methods, and also one laboratory that performed BESID and reported the correct result. However, there were a couple of labs that did immunohistochemistry and reported results. And one laboratory reported this case 2 as a positive by immunohistochemistry. However, they went on to look at it using NGS and didn't detect a fusion. Now, we decided that we wouldn't assess IHC results for this because we hadn't validated any immunohistochemistry, and because it's a different basis of the test, we didn't think it was appropriate to assess it. In this case, Helen mentioned to you that the amount of neoplastic cells in a tumour is crucial as to whether you're going to get a good reportable result or not, and nearly a quarter of the laboratories failed to provide a statement on the neoplastic cell content of the sample. So some more general comments. We noticed that laboratories weren't always using the most up-to-date references to support results, and the references really should be used to support the results, particularly when it's something quite new like this. If you use genomic coordinates in the report, you should include the genome build. Again, I want to emphasize about reference sequences, particularly where fusion is detected and you specify either nucleotide or exon position. You do need to include a reference sequence. However, if you don't find anything, you don't need to include reference sequences for all the genes on a large panel. This will just be can make the report too big. And as for all our EQAs, HGBS nomenclature should be used with appropriate IE if you find fusion and if your technology enables you to describe the fusion using HGBS. Now, HGBS for fusions, however, has only recently been introduced to the HGBS family, and it was a release in May of this year that includes the correct nomenclature to describe RNA fusions, which was one of the reasons that we didn't assess this as part of this EQA, because we appreciate it is quite new. So RNA fusions are described using the, I don't even know what you call that, two sets of dots to indicate the fusion. Use numbering according to the coding sequence of the transcript, with position 1 being the ATG codon, so not the start of transcription. This is, we notice that quite a few labs actually use the transcription start as position 1 of the nomenclature. If you've only analysed the breakpoint and not the entire transcript, you can use a question mark to describe the start and end points because they haven't been fully characterised. And that may be the case in quite a few of these fusions. So in case 1, the fusion can be described as what I've written here. So it's, we don't know where the start point of the start is, but we know the breakpoint's at position 1009 according to NN 001987.4, and then it's fused to position 1586 of the other NN 0010123338.2. Again, these are really important. So moving on in the future, we will be looking for laboratories to use this HGBS because it's quite important that everyone's using the same nomenclature. And this gives a way of seeing where the breakpoints are. And as Helene says, that's going to become more important in determining whether fusions are pathogenic or not. So, as I mentioned, we had designed this EQA to be done using RNA or RNA-based technology or NGS. So that could be DNA or RNA-based NGS. And as we'd expect, the majority of laboratories actually did use NGS, with most laboratories using RNA-based NGS. And there were a number of laboratories. The most common methods used were either various Archer fusion panels or on-combined focus. And again, there were also labs using various Illumina panels as well. There were 2 laboratories who used DNA-based methods. At least one of them was a capture-based method. And then there were laboratories, there are various commercial kits now which are available, which are either based on basically RNA-based methods, so things like NanoString, or multiplexed RT-PCR type assays. And we had 4 laboratories using these methods. In addition, I mentioned we had 2 laboratories who used IHC and 1 laboratory who used FISH. The laboratories using the immunohistochemistry actually used it in concert with NGS, and because these samples had actually been identified originally by FISH, we did assess the FISH part of this EQA. For next year, we're going to carry on this pilot EQA, because I think it was quite successful. There will be 3 cases sent out next year. We'll also allow laboratories to submit results using FISH, because we do appreciate that there are a number of laboratories are doing this, and we have had some interest in that. And we'll also look at the interpretation of the results for next year's EQA. However, it will still be a pilot EQA, so there won't be any performance associated with the EQA. So just to thank you for listening to this, but also I want to thank all the people who help us with the EQAs. That includes our specialist advisory group for molecular pathology, our group of experts who advise us on EQAs, our assessors who are experts, and Mark the EQA returns, and the sample source and validation laboratories who we wouldn't really be able to run the EQAs without, because we need to make sure we can get samples and get them tested and validated to make sure they are, and most of all our participants who we wouldn't be able to get by without. So thank you very much for that. And I'm going to pass you back to Sandy now.
Great, thank you, Jenny, and both thank you to you and Helene for a really interesting session covering all the elements of the EQA setup and format, the NTRK testing strategies themselves, and also then the feedback on the NTRK pilot. So we've got a couple of questions coming in. First of all, Helene, I'm going to come to you. So you described all the different technologies and the pros and cons of each. So somebody here is asking, why should labs perform NGS for NTRK fusions when IHC is quicker and cheaper?
I think it partly comes down to the information that you get and the sensitivity. The NTRK is much more sensitive, and it also picks up these novel fusions. We know that the IHC has potentially got some false positives and negatives on it. You're just directly looking at protein levels, whereas the NGS assay gives you much more information about what's causing that and the fusions, and allows you to interpret it much more. And it's, as I say, it's a more sensitive test.
Great, thank you. So basically getting more information for your single tests then?
You are, yes.
Yeah, great, thank you. Jenny, I know you mentioned about NTRK pilot continuing next year. So when will the next EQA run be available for NTRK?
We're going to be sending out next year's EQA towards the end of August. But if you're interested in participating in next year's EQA, enrolment is now open on our website for all our 2021 EQAs, and we very much appreciate your participation in them.
Great, thank you, Jenny. Somebody's very keen, which is good to see. Helene, back to you. So quite a good question, this one. Should labs set up NGS for DNA or RNA-based testing for NTRK fusions?
I think I personally would recommend RNA-based approach. We know that from experience of looking at whole genome sequencing data, that you pick up an awful lot of rearrangements with DNA, but it's very difficult to get information about whether they will be important fusions, whether they're going to be oncogenic, and whether they're going to be actual fusions that affect protein levels, whereas the RNA does give you a much better indication of whether it's a true fusion and affecting protein levels, and therefore clinically important. So I think the information you get from RNA is much more realistic of what would be happening in the cell, whereas the DNA, there's so much structural information going on there, it's not really an indication of whether it's an actual oncogenic fusion.
Great, thank you. That's very clear. We've got a question that says a bit more about the treatment options. So I'm not sure if we can, and licensing, so I'm not sure if we can cover this, but do we know if patients will still be offered NTRK inhibitors if the fusion partner is not identified? Helene, have you any background knowledge on that?
As far as I'm aware, the license is to do with the NTRK side of the fusion. So long as it's a proven oncogenic fusion, it doesn't matter what the partner is, and it doesn't particularly matter whether it's identified or not. It more comes down to being able to interpret the NTRK fusion data as to whether it truly is going to be clinically relevant.
Great, thank you. Jenny, can I come back to you? So how do you choose which samples and cases, I suppose the clinical case scenarios, to be used in EQAs?
So we look at, and on one hand there is what we can find, what's available, but we try to reflect the clinical situation as much as possible. So we would include common mutations quite often, more often than rare mutations, although we would obviously put rare things in there occasionally, because we're trying to test both what people do on a day-to-day basis and what we'll do with something that's novel and different. But also we look for cases where there is no fusions present or no mutations present, because it's important for the reporting and determination of whether people are picking up false positives in tumour testing, to include them in our EQA. So we try to get as much a broad spectrum of different cases as we can, and we try to reflect what laboratories are really getting in terms of the cases, and also look at case scenarios, ones that are relevant to particular situations. So in this case, in these cases of NTRK, we were using examples where there was a real possibility that they would be tested for NTRK fusion. Given the current situation, in future that would probably be much more broad-based, but we have to try and map it towards what the availability of drugs and the sort of recommendations of the one given a prescription of drugs is as well. So we also liaise with our expert advisors to help us choose these cases. So it's not just me that chooses the cases, I do it by speaking to the experts in the laboratories who are testing on a day-to-day basis.
Thank you, Jenny. And I guess just to add, also it depends what's available, isn't it, as well, particularly as we like to use real patient material.
Yeah, absolutely. And we're always looking on the lookout for interesting things to share with our participants, interesting cases.
Great, thank you, Jenny. We've got another interesting question here. So Helen, back to you. Is there any data around what percentage are novel gene fusions? Now, it might be a bit too presumptive to know that with accurate percentages based on the fact that we haven't been testing for gene fusions by NGS for very long, but have you any feeling of what that percentage of novel fusions are?
I'm afraid I don't, unfortunately. I think, as you say, I think we're too early in the testing, especially we're too early in testing using technology that can detect these novel fusions and define them. So obviously we can tell you what fusions we have identified and we know what's out there, but we don't really know what proportion we're going to find that are new and novel. So no, sorry, I haven't got any feel for it at the moment.
Great, thank you. I think that's something that is really important to share nationally, in particular across the UK, so we can actually get that information collated and look for publications.
Yeah, I think it'd be really interesting to review all the NTRK fusions after a few months, a year of this service, with everyone using RNA and NGS platforms.
Yeah, absolutely. There's more questions coming in. I can't quite, let me just try and find this one here. Right, so, but Jenny, HGFS nomenclature, so it might be difficult, might be the best documentation, but very difficult to interpret and relate to literature. I think this is in the field of fusions, coming into more of the molecular HGVS variant nomenclature. Have you got any comments on that and the approach that the assessors took for these EQA reports?
Yeah, so I think in both this EQA and other fusion EQAs, we appreciate that this is new nomenclature and there won't be that much available in the literature necessarily to relate to it. However, I think it's at the moment it's perfectly acceptable for laboratories to give exon numbering on their reports as well, and use other types of nomenclature as well, because agreed it might not be the easiest for people reading the report to understand this nomenclature straight off, your clinicians and stuff, until they become more familiar with it. So I think the best approach is perhaps a mix of the two, and we've often said that that's acceptable in other areas as well, where we think that you should use the HGVS, but if you feel that it requires more clarification, you can describe it in different ways as well, and that's acceptable as long as it's what you're describing is correct.
Thank you, Jenny. So I guess the message about the standardisation of nomenclature is to help drive comparable data sets and in biobanks and databases, so we can actually compare the same data regardless of which lab's doing the testing. But obviously on the flip side, the reader of the report has to understand what that result is very clearly as well. So it's getting that fine balance, isn't it?
Absolutely.
Yeah.
And I think it's fair to say that's true for a lot of fusions, and until the literature becomes available, it's true it's going to be hard, but hopefully by using everyone using H2EX, it'll be much easier for people to compare their results.
Yeah, absolutely. Okay, so I think we will close the session now. Jenny, I'm going to come to you for some feedback. We've also had a couple of people asking if the slides can be shared, which comes on very nicely to what updates you're going to give us now.
Yes, so once this webinar is over, in about an hour's time, you'll be sent an email from the GoToWebinar, which will include a link to a survey, which we'd very much appreciate you answering if possible. And we very much appreciate any feedback on our webinars. You'll also be able to view this webinar for the next month on the GoToWebinar website using the link that you received. And they'll also be available more openly on YouTube after this month is over. And if you want to register for our YouTube channel, you can do, and you can subscribe with it. In the email you get from the GoToWebinar, there will be a participation certificate included on your email as well. And we're trying to offer our participants a lot more of these focus on topics. And our next one from November is going to be on newborn screening with a particular emphasis on severe combined immunodeficiency. And thank you for all attending.
Thank you, everyone, and thank you both to Jenny and Helene for a really interesting session. Thank you and goodbye.